Ever wished you could just hit pause, on everything?
On life.
Yeah, like everything, even down to like the cellular level.
Ah, I see where you're going with this.
Well, that's basically what we're diving into today. Cryopreservation.
It's pretty wild, right?
Totally. Like
Ah,
putting cells in suspended animation.
exactly. And that's such a valuable tool for research, clinical applications, you name it.
Oh yeah, there's a ton of applications. Like you can preserve those really rare cell lines.
Oh, for sure. Or think about genetically modified cells.
Right. It's like having this living library of cells whenever you need them.
Yeah, exactly. It's not even just like having them on hand. It means you can do these experiments with the same cells over and over.
So you know your results aren't messed up by having slightly different cells each time.
Exactly. It's huge for getting consistent and reliable data.
That makes a lot of sense. So how do you actually freeze cells and then bring them back?
Well, it's a whole process. First, you have to get the cells ready.
So like harvesting them from wherever they're growing.
Yeah, and then you concentrate them.
Makes sense. What's next?
All right, here's where it gets interesting. You have to add this cryoprotectant.
A what?
A cryoprotectant, usually it's the stuff called DMSO.
Okay, and that does what?
It protects the cells when you freeze them.
Like a little shield.
Pretty much. It keeps those ice crystals from forming.
Ah, because ice crystals would be bad.
Really bad. They can like totally wreck the cell membranes like tiny little daggers.
Ouch. So the DMSO stops those daggers.
Exactly. And then comes the freezing part. You got to lower the temperature really slowly.
Like how slowly are we talking?
Like 1 degree Celsius per minute. Slowly.
Wow, that's pretty gradual.
Yeah, and you need special freezers for that, or you can use these alcohol-based containers,
So there's like a high-tech and a low-tech way.
basically. But either way, you got to go slow and steady.
Got it. Slow and steady wins the race. And where do the cells go once they're frozen?
Into liquid nitrogen.
Whoa, liquid nitrogen. That's serious.
Yeah, it's super cold, like minus 196 Celsius.
That's insane.
Yeah.
So everything just stops at that temperature.
Pretty much, yeah.
Yeah.
All biological activity just grinds to a halt. It's like the ultimate pause button.
So they can be stored like that basically forever.
Yeah, pretty much indefinitely, as long as they stay in the liquid nitrogen.
That's wild. Okay, so cells are frozen, everything's stopped. How do they survive that? What's happening at like the molecular level?
Well, their metabolism basically goes way, way down, almost stops completely. And the cryoprotectant, that DMSO we talked about, that helps a ton.
So it does more than just block ice crystals.
Yeah, it helps with something called vitrification.
Vitri what now?
Vitrification. It's kind of complicated, but basically the cells turn into this glass-like state instead of forming ice.
Oh, wow, that's pretty cool.
It is, and that helps protect them even more. And that's super important for preserving things like organs, where you have all these complex structures.
Right, got to keep all those delicate parts safe.
Yeah.
So you've frozen the cells, everything's on pause. How do you bring them back to life when you need them?
So now you got to thaw them, and this part is crucial. You got to do it fast.
Really? Why fast?
Because if you thaw them slowly, guess what happens?
Uh-oh, those ice crystals come back.
Exactly. So we got to avoid that at all costs.
Gotcha. So how do you thaw them quickly?
The most common way is to use a water bath, like a warm bath, at 37 degrees Celsius.
And you just dunk the cells in.
Yeah, you put the vial in there and kind of swirl it around. But you got to be careful. You don't want to leave them in too long.
So it's a delicate balance. Got to be quick, but not too quick.
Exactly. It usually only takes a minute or two.
Okay, so they're thawed. What's next?
Right after they're thawed, you got to get rid of the cryoprotectant, that DMSO.
Oh, right, because it was only there to help with the freezing.
Exactly. If you leave it in there too long, it can actually become toxic to the cells.
Yikes. So how do you get rid of it?
You basically wash it away. You transfer the cells into fresh media, kind of like giving them a bath.
And that gets rid of all the DMSO.
It dilutes it out so it's no longer a problem.
Mm.
And then, you got to be really careful with the cells, handle them gently.
So they can recover from their frozen adventure.
Exactly. You got to give them the right environment to bounce back and start doing their thing again.
It sounds like every step in this process is super delicate. Are there any challenges that, researchers come up against?
Oh, of course. Not all cells survive the freezing and thawing, unfortunately.
So some cells are tougher than others?
Yeah, like some primary cells or certain stem cells, those can be a bit more sensitive.
So it's not a one-size-fits-all kind of thing?
Nope, not at all. And then there's always the risk of that cryoprotectant toxicity we talked about.
Right, if the concentration's off or something.
Exactly. And of course, there's always the chance of contamination, especially when you're thawing the cells. You got to be super careful.
Sterile technique is key.
Absolutely. You're dealing with the building blocks of life here.
Oh,
Got to be precise and careful.
makes sense. So, for someone who's thinking about trying cryopreservation, what are some key things to keep in mind?
Well, first off, make sure you freeze the cells at the right rate.
So that slow and steady thing we talked about.
Yep. If you have access to a controlled rate freezer, that's ideal. But if not, just make sure you create a controlled environment. No chucking them in the freezer haphazardly.
It's all about respecting those tiny cells.
Absolutely. And make sure you use the right cryoprotectant. DMSO is a good go-to for a lot of mammalian cells, but there are others out there.
So do your research first.
For sure. And like we talked about, be super careful with the thawing process. Be ready to dilute that cryoprotectant right away.
So be prepared. Have everything ready to go.
Exactly. And handle the cells with care every step of the way. Don't jostle them around. They're delicate little things. And last but not least, check their viability after you thaw them.
So a final checkup to make sure they're okay.
Exactly. Make sure they're happy and healthy before you go using them for anything.
Awesome. So it's all about setting those cells up for success.
Exactly, whether they're going into storage for research, clinical applications, or even, who knows what in the future.
The future of cryopreservation.
It's pretty exciting to think about. It's like they get a fresh start after their time in the freezer.
I love that. So we've talked about how cryopreservation works, but why should anyone care? What's the big deal?
That's a good question. It's not just some weird science experiment. Cryopreservation is actually super important for tons of different fields.
Like what?
Well, think about it. If you can freeze and store cells, that opens up so many possibilities for research, medicine, even things like conservation.
It's like a time machine for cells.
Exactly. Researchers can use cryopreservation to study cells from different points in time or under different conditions.
Oh, that's cool.
Yeah, it's really changed the game for fields like drug discovery, toxicology, just basic cell biology research in general.
What about medicine? I bet this is useful there too.
Oh, absolutely. It's essential for things like bone marrow transplants.
How so?
Well, they take healthy stem cells from a donor, freeze them, and then transplant them into a patient later on.
Wow, that's amazing.
It is. And it's also used in fertility treatments, freezing eggs and sperm for later use.
Right. So this isn't just about cells in a dish. It's actually helping people in real life.
Exactly. And it goes even further than that. Scientists are looking at ways to use cryopreservation to preserve whole organs for transplant.
Whoa, really?
Yeah, that could totally revolutionize organ donation.
That's incredible. It's like something out of science fiction.
I know, right? And if you think even further ahead, cryopreservation could be important for space travel.
Space travel.
Yeah, think about it. Astronauts could bring important cell lines with them on long missions, or even tissues.
Wow. So the possibilities are pretty much endless.
Pretty much. As we learn more about cells and technology keeps getting better, who knows what we'll be able to do with cryopreservation?
It's definitely a field to keep an eye on. This has been a fascinating deep dive into the world of cryopreservation.
I know. It's amazing to think about all that potential.
Yeah, it's like we can really control life at the tiniest level,
It really makes you think about what's next for cryopreservation, especially as we learn more and more about cells.
Oh, absolutely. But, with any powerful technology, you have to think about the ethics of it all, too.
That's a good point. We're kind of entering uncharted territory here.
Exactly. There are a lot of big questions to consider, like what about consent? And what about storing these cells long term? What does that mean for future generations?
Yeah, it's a lot to think about.
Definitely. It's not just about the science. It's about how we use that science responsibly.
It's like science, technology, and ethics all coming together.
Exactly. So as we wrap up our deep dive into cryopreservation, is there anything you want our listeners to really take away from all this?
That's a good question. I think the biggest thing to remember is that cryopreservation is about more than just freezing cells. It's about manipulating life itself. It's about time and what it means to be alive.
Whoa, that's deep.
It is. And as we keep exploring this technology, we're going to keep making incredible discoveries, but we're also going to face some tough challenges.
That's a great point to end on. It's like science isn't just about facts. It's about exploring the big mysteries.
I like that.
Thanks for joining us on this journey into the world of cryopreservation.